The next step in the scientific method then is the experiment. We would be able to find out which of those hypotheses is correct if we do experiments. So to answer that, to figure that out, we have David Henry. You work at Padilla Bay National Estuarine Research Reserve. What is your job there?
I run a lab that we test fecal coliform, or E. coli, to find out how they are doing in the environment. Are there a lot of them? Are there not so many of them?
Aha, this sounds like part of our theme today, doesn't it? So, David, what was your observation?
Well, like Greg pointed out at the beginning, we observed that there were high fecal coliform levels here at Bayview State Park.
Did that lead you to a question?
It did. The research question we want to know is, why are the fecal coliform levels high at Bayview State Park, and where are they coming from?
That would be a good question to answer. Maybe we could get to a solution that way.
I hope so. That's the bottom line. Can we come up with a way to limit these so we can eat the oysters in the end?
Obviously, we haven't come that far yet, have we?
We're getting closer. Science moves slow, but we get there.
All right, thanks. And so now we have asked the question about where they are and why they're there. What is a possible hypothesis? What hypothesis were you working on with your experiment today?
Well, when I want to make a hypothesis, I kind of listen and do research. So I listen to what the students have been saying about their hypotheses, asking more questions, trying to figure out where it's all coming from. And then I gather information. So I looked at Andrew's map, where there's deep water and it's colder, and there's warm water close to shore, and maybe there's a relationship there. Because we know from the past that fecal coliform live inside warm-blooded animals. They like warmth and heat. So my hypothesis was, out in the cold water, out in the deep part of the bay, you're not going to find as many fecal coliform. They like warm, so they're going to be closer to the shore.
That sounds like a prediction.
That's what that is. I hope so.
All right, so then you were able to design an experiment to address your hypothesis.
Exactly. Yesterday, Andrew went out in his boat and he collected 3 samples from out in different parts of the bay, way out in the deep water where it's cold. And then meanwhile, I was on shore and I collected 4 samples along the shore at different places.
All right. Can you show us how you do your experiment, please?
Well, I'm going to need some help here. I need Pat and Steven. These guys are experienced fecal coliform testers. And what we did is we went out in the bay and we scooped, can you guys show them the scooper? We scooped samples in a jar where we could take about 250 milliliters of water. Now the state standards and all around the country, they judge fecal coliform for how many are in 100 milliliters of water. So you guys, how did we get it into 100 milliliters of water? How do we figure that out?
We poured it into a granulated cylinder.
Right up to the 100 number. Excellent. Then what are we going to do next?
Gonna pour it into the jar.
Yep.
Steven said you're going to pour it. Tell us again, Steven.
We're gonna pour the water into the filter area so we can get, see how much E. coli there is.
All right. There goes 100 milliliters of Padilla Bay water into the cylinder. Okay.
Next, I get to pump the water down into the bottom.
Okay. Pump away. Steven, what do you see happening down at the bottom of this pump system he has going here?
The water's going through the piece of paper into the bottom of the cylinder thing.
I see. You see there's a piece of paper in the middle there?
Yes.
Okay.
And Alex, that piece of paper is a special membrane, kind of like Gore-Tex fabric on rain jackets, which we in the Northwest know a lot about. And that has small holes in it, so it's designed to trap exactly the same size as these bacteria. So we've got a special paper here, and it's 0.45 microns wide. That's like a fishing net that's 0.45 microns wide, very small.
Very small. So you're filtering the water. Speaking of filter, the oyster is a filter feeder. They filter plankton. Do you think they might catch some of these bacteria too?
What do you guys think?
Yeah.
Oh yeah.
Oh, okay. So there's the filter. We've got the filter paper ready now. What's next?
Steven, you want to show them?
We're gonna put this dye onto the paper so it covers it up, and then we're gonna slide the piece of paper onto the petri dish.
Okay.
So go ahead and put that in there. This is kind of like a Denny's all-you-can-eat 24-hour banquet here. This is a lot of food. The fecal coliform are gonna love it. They're gonna eat it, grow in it, and hopefully we can get the paper onto this broth without contaminating it, because we want to do it in a sterile fashion. So how are we gonna do that, guys?
Get this part on fire.
So that's good. You can't see it, but it's lit. We've got a tweezers. We're gonna dip it in some alcohol. You're gonna hold it on the flame. That's gonna burn it off. Now we're gonna see if we can grab that paper off there. Excellent. This guy is good. Even in the wind. And putting it down there with the tweezers. Great. Then go ahead and get the lid back on. There we go. So those bacteria are ready to go. They're on the paper. They've got a nice shelter. They've got food. We're going to stick them in an incubator for 24 hours where they can get warm and grow. And just like any good cooking show, we've done that ahead of time so you guys don't have to wait 24 hours. And we have a tray over here if you want to come and look at it.
So you have results. Heather, that's the next step, isn't it, of the scientific method. So Heather will bring over our explanation of the results. That's right, that's where we decide what it means. And here on the table we have results. David, are these students prepared to explain the results?
I might be able to do that because I don't want everyone touching these. These are E. coli bacteria growing in here, fecal coliforms, and so I don't want people with gum. Whenever we're working around this stuff, we definitely wash our hands a lot and we're really careful with it. But you can see right on this tray where we have about 89, we go in and we count every one of these blue dots, and that's a bacteria that's grown over the past 24 hours. And this sample came from one particular place in the bay out of our 7 places we tested. Okay.
Okay, that's the little dish there. What else are you going to show us, David?
Well, I'd like to show the map here just to give an idea, because I know people are from around the country, they don't know this area. We are situated right here. There's a little town on that map called Bayview, and we're right by the number 3, and that's Bayview State Park. And the 3 is the result when we went back and counted the colonies or number of bacteria that grew in 100 milliliters of water. It was 3. Below it, you'll see right off the town of Bayview, we got a count of 12. Then another shallow site along the shore down by this slough or river coming into the bay, we got 3. Out in the bay, we got in the deep cold water where Andrew was at, we've got 0, 0, and then way up to the north we got another 0. And then the last result was the 89, that one I showed you. We got a really high count by that river coming in over there. Those are our results right there.
That's very interesting results. Thanks, David. I bet we can start to learn a lot about the locations of the fecal coliform bacteria in the bay.